It all sounds so much more impressive when Lila says it, doesn't it? Hi, friends. Thanks so much for staying with us today. I know it's been a long day. It's been an exciting day. We're bringing it home now at this session, and then we have a couple of really more interesting things going on. So today we're going to take a deep dive together into this thing called CRISPR, which is a very popular buzzword these days, and there's a lot going on in the news, a lot. So what I did when I was putting this talk together for you was try to think about what should most interest you in watching this field and what's going on right now and in what's moving forward. And this field is moving forward so quickly that even I am astonished, and it is my job to follow this field and to follow fields that move fast. So quickly, in fact, that one of the laws we're going to talk about today changed while I was making this presentation. Like literally, I Googled it once, I Googled it an hour later, and the law had changed. So that's why we're going to take the time together and really look at this space to think not just about where we are now, where can we be in the future, and where do we want to be in the future? And that's how we're going to be rethinking the future of health. So Habib did a really great job setting this up so we can move a bit quickly through our bio reminder. But what is digital biology? So as Habib pointed out to you in the last session, we've been able to digitize the information of life. And one of those molecules that we've been able to do very rapidly, cheaply, and exponentially is DNA. So just as a reminder, all life on Earth is related. All life on Earth is made of cells, and all life on Earth runs on the same operating system. So if you take, let's say, your body, and you zoom into a cell, and then you zoom in even further, eventually you get to the collection of DNA that is like the operating manual for you. Those are your chromosomes. Do you guys remember how many chromosome pairs do humans have? 23, very good. There's a famous company named after it. Right. So then if we take one of those 23 chromosomes and we zoom in even further, we get all the way down to the level of the famous DNA double helix, which is made up in natural systems of 4 chemical building blocks that, for short, we call A, C, T, and G. Right? So that's what DNA is. Your body, you have about, let's call it 3 billion base pairs of DNA, and 99.9% of your DNA is identical to the DNA of every other human on Earth. I'm going to say that one more time because that thought helps me get up in the morning when I've been listening to the news the night before. Every human being on Earth is 99.9% identical to every other human being. So all these reasons we kill each other, we make that up, right? It's not hard-coded in our DNA. So computers store information as zeros and ones. Life stores information as A, C, T, and G. And this is important because if you use this paradigm of digital biology to think about life as information, now you can see it the way I see it, which is when I look at a DNA helix, which I do, I'm a nerd, I don't actually see what I'm showing you here. I see this. I see code. I see information. And this is an important paradigm because what it means is that anything that we can do to computer code, we can also do to DNA code. So let me give you some examples. If we wanted to move big blocks of code around, that would be genetic engineering, which we've known how to do since the 1970s, right? Let's say what I want to do is write DNA code from scratch. So that's synthetic biology. We had a really awesome lunchtime panel about synthetic biology. But let's say what you want to do is go in and debug the code of life. Change precisely and permanently, one letter at a time, the DNA sequence. That's what's called gene editing. And that is one of the most powerful technologies that mankind has at its disposal right now. So let's take a look real quick at what this thing CRISPR is. It's the elephant in the room. So CRISPR is not something that humans invented. It's actually the bacterial immune system that evolved on Earth many billion years ago. We just figured out how to hijack it. So the word CRISPR is an acronym for a pattern of DNA that is part of this system. And I'm not going to bother telling you what it stands for because you won't remember anyway. It's not important. But here are the pieces of how CRISPR works. First, there's a protein, and that protein is called Cas. And there are different flavors of Cas that have numbers after them. So the most common variant that we see today is called Cas9. And you can think of Cas9 like a hand, okay? So one of the things that that hand grabs onto is a molecule which is called a guide RNA. And what I'm showing you in blue here is that piece of guide RNA is designed to have a sequence that you're interested in. So here we're calling that the blue sequence. And then what happens is Cas is holding onto that guide RNA, and then it goes and it binds onto the genome of the living cell. And then it runs along that genome until it finds a place on the genome that is an exact match to that guide RNA. And so now you can see it's holding 3 pieces, just like I'm showing you in the picture here. And when Cas finds that perfect match, then it turns into a pair of molecular scissors. And so it cuts the DNA. And what you get is a broken piece of DNA like this. Now, living cells really, really don't like broken DNA. So there's a whole system of DNA repair mechanisms that you have in your body and in every cell. And that's what the cell uses to try to glue together that piece of DNA. Now, here's where the human comes in CRISPR. Let's say that you don't actually want to keep that AAA that was in our original genome. We want to make one of those A's a C instead. So here, what I do is I trick the cell when it's trying to make the repair of the DNA. I give it what's called a template that has the sequence that I actually want, the edit. And then when the cell very happily makes that repair happen, it goes ahead and it puts in that C, which is the edit that I wanted instead of the A. So that is very, very basically how this works. It's just hijacking the systems that the cell already has. So there's lots of technical detail here, and if you are as excited as I am by the stoichiometry of non-homologous end joining to resolving holiday structures, please come find me, because I would really like to talk to you. But we're not going to go deeper on that stuff today. Instead, I'm going to move us into a bigger discussion of why this technology is so important. So it has vast applications, whether we're talking about agriculture. For example, this is, I think, one of our greatest tools to fight climate change, is this system. So whether we're talking about agriculture or environmental or monitoring or education or even health, CRISPR has massive, massive applications. And a really simple one that I can tell you about in healthcare is a really cool one, which is just diagnosing. Remember I showed you that CRISPR acts like a programmable pair of molecular scissors. It has a fantastic search function. So a company called Sherlock Biosciences has used a different flavor of Cas. It's called Cas13. And they figured out how to make guide RNAs that match a disease. And then what happens is, in this system, which is also called Sherlock, when Cas13 finds the disease RNA that it's looking for, it cuts, right? That's what Cas does. But then this particular variant does something special. It becomes supercharged, and it goes on and it cuts more molecules that are what are called reporters. So I'm showing you those in pink here. So when Cas cuts those reporters, they make a signal. And so what Sherlock has done is put 4 of these signals onto a strip of paper. So essentially what you get is like a pregnancy test for 4 diseases simultaneously, looking for the RNA of that disease. Now, this is remarkable because this is a paper-based test, which costs 60 US cents per test, right? That's 50 euro cents per test. That is spectacularly cheap and spectacularly sensitive. So keep an eye on things like this. But this is actually not why we're here today to talk about CRISPR. We're here to talk about the complicated stuff, like this. So you probably remember in late last November, there was a firestorm set off when MIT Tech Review broke the story that a scientist in China had edited the genomes of 2 baby girls, and those 2 girls had actually been born. And when this hit the news, it exploded. It hit the mainstream news, and also the scientific news, it just blew up. Because out of nowhere, suddenly we had 2 baby girls on this planet whose genomes had been CRISPR'd. And this was announced on YouTube, not in the scientific literature, which isn't how we do things. and was also in flagrant violation of a variety of laws and also ethical concerns. And so what I want to do for our rest of our time together is walk through both this story of, their names are Lulu and Nana, the two baby girls who were born, but also use a framework to look forward as to how we can take this tool, which has the potential to be one of the most impactful things we have for human health that there's ever been, but how we make sure we end up in places we actually want to be. Okay, so here's the framework we're going to use today. It was a framework that was used by some Mexican researchers who were using this actually to edit mosquitoes. And they ask 4 questions, and they suggest that we ask 4 questions. The first question is there a need to do this CRISPR intervention for health? The second question is, can it be done? That's a technical question. I'm a scientist. I really like technical questions. The third question is it legal? And the fourth question we'll look at is, should it be done? Which, of course, is where things get really interesting. So is there a need? That's our first question. That is a public health question. So let's take a quick look at the types of diseases that we could actually use CRISPR for. So it turns out that genetic disease comes in a variety of flavors. One of the flavors is when you have too many or too few chromosomes. So, for example, Down syndrome is a perfect example of that, right? Trisomy 23. Or you can have too many copies of a big chunk of DNA. Frequently, these things cause cancer and other type of neurological diseases. Another type of genetic disease is what we call a Mendelian disease, which means it's a disease that arises from one gene and most likely one place on one gene. So a single gene causes a disease. And the third major flavor of genetic disease in humans is what we call multifactorial, which basically just is a fancy way of saying complicated. It means there are lots of places on the DNA that contribute to a disease, but also your environment matters. Where you eat, whether you smoke, how dirty the air is, how dirty the water is. All of those things feed together in a complex way to make your disease happen and to make it look a particular way. So CRISPR, at least the way we have it now, is most effective for this type of disease, for a single gene, a Mendelian variant. And why? And it's because this is a case where we know the edit that we need. We know the part of the DNA that's causing the disease, and we know how to change it to make the disease go away. Now, there are only about a few thousand of these types of Mendelian disorders. So there's a few thousand places on the genome that we know causes these changes, very specific. And then there's a few hundred phenotypes, some of which are disease, that come from these changes. And most of those are really very rare. However, some of them you've heard of, right? So I'm talking about things like, this isn't a disease, but color blindness, for example, is one of these single gene changes. And then other diseases that come from these single gene changes are things like cystic fibrosis, sickle cell disease, beta thalassemia, a few other of these types of blood diseases also come here. So this is where the low-hanging fruit is in this innovation space, and that's where we're going to be spending a lot of time talking today. Now, there's another distinction that I want us to be able to draw because it also affects how we view using CRISPR in humans, and that is, where exactly are you doing the edit? So let's say, for example, that I'm interested in editing a skin cell or a group of skin cells on me that has a tumor, right? I could use CRISPR, just like Habib told us. Those cells have accumulated a mutation or multiple mutations, and I could use CRISPR like the eraser on a pencil to dial away those mutations and put my skin cells back the way they were. That's called a somatic edit. Soma from the word meaning body, right? So I'm just changing my skin cells. If I were trying to edit my heart cells to get rid of a heart disease, or I was trying to edit my lung cells to get rid of cystic fibrosis, or my blood cells to get rid of beta thalassemia, those would all be somatic. So let me ask you, if I make a change to my skin, does that change get passed on to my son? It doesn't, right? Because all I've done is change my body. All I've done is change my skin. In contrast, if I were to edit my eggs or my partner's sperm, then we have now made a type of change which is called a germline change. So would our son have edited a change from the sperm, or inherited a change in sperm or a change in my egg? Yes, absolutely. And so this is where things get complicated. And this is also what happened in China when these baby girls were edited as embryos during IVF. Because now it's not just the baby girls that were edited, it's the eggs that are going to be growing from them as well. So they will pass these edits on to their children and their children and their children. So let's take a closer look at what exactly was going on in this scenario. And I'm going to show you this headline here for the last time, because I really want us to remember that what we're talking about is children. We're talking about 2 baby girls. This is not a picture of those baby girls. There are no pictures of those baby girls being shown publicly right now. And in fact, none of the data from these baby girls has been made public so that scientists could independently verify what happened. But I just want to remind us that we're talking about children. Because when you look online and you see people talking about CRISPR babies and, oh, this is the worst technology of 2018, MIT Tech Review actually wrote that, it broke my heart because we're talking about 2 little girls. And 30 years ago, people used to say, oh, children who were born by IVF, they're not even human. They shouldn't exist. Could you imagine saying that to a child today who was born by IVF? Right? It's too common. You would never say that. And so I just want us to remember that even though these girls had their genomes edited as embryos, they're still people. And they still deserve our protection and our love and our respect. So I'm going to keep showing us pictures of babies to remind us of that. All right, so of those diseases I told you about, cystic fibrosis, beta thalassemia, sickle cell anemia, which one of these diseases was the target here of the Chinese experiment? And the answer was none of them. He chose, Dr. He chose as his target HIV. So what he did, or what he claims to have done, because again, there's been no independent verification of this, was he took a gene which is called CCR5, and CCR5 encodes what's essentially the molecular door that HIV uses to get into your T cells. So if that molecular door is the wrong shape, or the molecular door isn't there, HIV can't get into a T cell. There are already people on Earth today who are immune to HIV because they have this deletion in CCR5. So what this researcher did was he took the eggs and sperm from couples, 7 couples, who were at a fertility clinic in China trying to do IVF, and the fathers were all HIV positive. So he claimed that by editing CCR5, he could make sure that those embryos would not pick up HIV during IVF. The problem with this, one of the problems with this, is that IVF, the way they wash the sperm, already gets rid of the HIV. So these embryos were never at particularly high risk of contracting HIV during that phase of IVF in the first place. So that's problematic. But we shouldn't undersell how much of a disease burden HIV still has today. It is still a serious threat. But remember, things like condoms and ART therapy has made a massive dent in the mortality of HIV, if not its spread. So this was disappointing to me because I would have loved to have seen the first test case in humans have been a silver bullet, that the only way to cure this disease was by editing the embryo. And unfortunately, that is not what happened here. And there were a whole bunch of other things that were sketchy about how things were done in terms of consent and funding and a lot of other things. But that's the one part I want to highlight. So going back to our question of is there a need, you could sharpen that question to ask, do you actually need to edit an embryo? Could you, for example, get the effect another way? Or is CRISPR the only way to do this? In diseases that are spread throughout the body and not just in one place, it actually could make sense to really use CRISPR and edit an embryo so that the change is in all the body cells. That also does not seem to have been the case with Lulu and Nana. Could you edit just specific cells? So Habib told you about folks who have had their eye cells edited. We'll talk about that again in a minute. David Roberts mentioned to you this morning that there have been successful trials in Canada where folks have their immune cells edited and then put back in their body. So could you just edit a particular part of the body instead of the whole body? And the last question that I ask is, does it really have to be done in the embryo? Could you treat a child or an adult this way instead? And so all of these are ways of looking at a need and looking for other ways that we could actually fix them, instead of taking a process that might be technically easy, but is ethically extremely fraught. Let's take our next step. Let's look at the technical question. Can it be done? So CRISPR, has anybody here actually done it with their own hands? Has anybody done an experiment? You guys, you gotta go play. So back in my day, when I was at graduate school, which, by the way, was not that long ago, thank you very much, we could do this stuff, but it was really hard. It was really, really hard. It took a really long time. Kids these days, they don't understand how easy they've got it. It's actually true. So, however, even though it's very simple, it's not necessarily accurate. So you might have heard of these off-target effects, which is a really, really big challenge with CRISPR, where once Cas gets activated and it can go and cut at the place you want, the scissors don't turn off. So they could be going around cutting in other places, and then you'd be getting edits in other places in the genome where you're not necessarily looking. And these are called off-target effects, and they are a big problem. And one of the major problems with not having Lulu and Nana's genome sequences being made available is that scientists aren't able to go in and look to see if there are other effects that happened. And in fact, there's a hint that the lead scientist in this study actually knew of at least one off-target effect and decided to implant the uterus, the embryo in the mother's uterus anyway. So what are we going to do about this? We really need more efficient CRISPR. And there are two basic ways to get that better efficiency. The first is to get a better pair of scissors. So people have been mining what we know about bacteria in order to find new flavors of Cas, and Cas that has different activities. So that's one way of making this better. A second way, though, is instead of looking for better scissors, look for better guides, right? And here's an article that we published on Singularity Hub not too long ago about this really cool experiment that a group at Duke did, where they figured out that if you actually change the structure of the guide RNA to put what's essentially a padlock on the end, then the guide RNA has to be an exact match in order for Cas to cut, and just making that simple change in putting that little molecular padlock on the end made CRISPR between 50 and 200 times more accurate. So that's huge. And in fact, I'm betting if you polled a lot of scientists and said, what's the one thing that makes you the most nervous about using CRISPR in the clinic? They would say off-target effects. So we're getting better. We're getting more accurate at how we do this. There are also folks who are trying to use AI in order to make editing more accurate. So if we're able to computationally predict better where those off-target effects might happen, then we could go look for them and make sure they're not there, and maybe even use CRISPR again to get rid of that off-target edit, right? Okay, and there are several groups that are working on AI approaches to better CRISPR. Is it legal? Now, here's where it gets interesting, right? The legal landscape on CRISPR is highly varied, in part because the technology is really so young, at least in human hands. So the very first report talking about this bacterial immune system was back in 1993, but the field didn't really heat up until 2011 or 2012, when folks at UC Berkeley and at Harvard and the Broad started realizing that you could move the pieces out of bacteria, put them in another system, and make them super duper powerful. So because this is such a new technology, there's a large spectrum in terms of its legality to use. So let's take the easiest case first, which is that somatic case we talked about, right? What happens if I just edit something in a person's body that can't be inherited? And that's very simple. It's not only legal in most places, these trials are going on right now. Habib told you about a whole bunch of them. There's a lot going on in China where people are taking immune cells out of a cancer patient using CRISPR to kind of arm them against the cancer and then putting them back into the cancer patient's body. There's just been approved another study at UPenn in the United States where now some American clinical trials are going to be happening. We've got companies like CRISPR Therapeutics and Vertex Pharmaceuticals who are working on blood diseases. That's one of those ex vivo cases where you take the blood cells out of the patient, put them in the lab, use CRISPR to get rid of the mutation, and then put them back into the patient. So that's also somatic. And then Habiba also mentioned this hereditary blindness use case. He showed you the picture of the young boy who's had those subretinal injections in order to cure his blindness. Well, now Editas Medicine has gotten approval to move ahead with an actual large clinical trial, not just one patient. So that's exciting because that's the first in-body application clinical trial of CRISPR, and that's just getting off the ground. Germline editing, though, is different, right? This is where you can pass the edit on. So, and there's a big spectrum here as well. In Canada, for example, a scientist can go to jail for making an edit in a cell that can be inherited. Okay? Actually, put your scientists behind bars. I was standing in front of some Canadian government ministers last year, and they were like, hey, what's the one thing we can do for Canada to be leaders in CRISPR? I was like, don't put your scientists in jail. That's your first step, guys. Let's look at the EU now. So the European Committee on Bioethics spent many years in conversation with a variety of stakeholders. And the results was, now look at the date, 1997. Okay, so we're just talking about DNA here. We're not even talking about CRISPR yet. And this is a convention called the Oviedo Convention. And here are the main tenets of that agreement in the EU. No enhancement is allowed. So we've only been talking about therapy, right? Curing a disease. But what if I could tweak your kid's DNA to make them a little bit stronger, or a little bit faster, or a little bit taller? Or maybe you don't like blue eyes, so you want brown eyes, right? Those are all enhancements. And the general community feeling is enhancements are not okay, but therapy is okay, diagnostics is okay. The Oviedo Convention also said no germline editing, so somatic only. And the Nordic region signed on, put into effect this convention with the exception of Sweden that signed but didn't ratify or enforce. But at least this rule is on the books. Another example is UNESCO had a bioethics committee that put together something with a very long acronym. And what that said was they called on the community of people around the world and said, no germline edits, no acting alone, and you must engage in broad dialogue before you use CRISPR in the clinic or in any sort of human health way. And so you can already see where poor Lulu and Nana were not part of that, right? We had an investigator in China who acted alone and possibly did not correctly inform the parents of what they were signing up to. So when the CRISPR babies announcement came out, there was a huge ethical storm, and ethical investigations were launched into a whole bunch of people involved, including the American scientists who knew about it while the experiment was going on. And those 3 scientists in America have actually all been cleared because they technically didn't break any rules, right? There's no science police. There's no science jail. So who were they supposed to tell? There was no one to tell. Science traditionally has been a self-regulating community. We've been pretty good for the last 1,000 years at saying what we're okay with and following the rules and like punishing each other when somebody breaks them. That system failed us here. So instead, what we're seeing are bodies like the World Health Organization putting together expert panels that are calling for rigor and transparency and total clarity before anybody gets to do any of these experiments, move forward in any of these clinical trials. Somebody has to know, and now there's someone to tell in case you hear something. So if you see something, say something, right? Now there's someone to tell. And in fact, while I was making this talk for you today, China came out and said that they were going to update their civil code to make an actual, to make doing this type of experiment the scientist personally responsible for what they're calling the personality right to have your DNA be protected. So China is also now taking a harder look at their regulatory system and their ethical system and saying, this was broken, this didn't work. And that's where we get to our 4th question. Should it be done? Because this is the million-dollar question, right? Who gets to decide whether an edit should be done to an embryo or to someone who's older? Now, if I were to do it to myself, I could make that call, right? But so one of the common arguments here is, well, the embryo doesn't get to choose. So how is it okay for the embryo to be edited? Now, I don't know about you guys, but I did not choose my DNA. Did you choose your DNA? None of us chose art. This joke is more awkward when my parents are in the room. I'm glad they're not with us today. It was really embarrassing one time. But okay, so in my opinion, no embryo got to choose its DNA. So I'm not really sure that's the argument we should be taking for should it be done. But there are other really important questions we need to be asking. So for example, are we talking about therapy or are we talking about enhancement? The global consensus right now from the scientific community is that therapy is okay, enhancement is not. Somatic or germline? Hopefully I've convinced you that the somatic case is pretty easy, right? Because it doesn't get passed on. But the germline is where things start to get really complicated, and that's where Lulu and Nana came onto the stage. And then there's another piece here which is about unintended consequences. Let's say we get all of those off-target effects taken care of. CRISPR's 100% accurate, and still we've made an edit that a human has designed. How sure are we really that that edit is doing what we think it's doing, and maybe not doing that plus something else? So you might have seen a headline like this about these two little girls, where it turns out that there were studies in mice that showed that CCR5, the target of this CRISPR, actually enhances memory function and memory storage in mice. So that wasn't what this researcher said he was doing. He said he was trying to keep out HIV, but what if these girls actually have better memories now? So that's not a terribly bad effect, but was it really unintended, and where do we draw the line, and who gets to decide? There's also been hints that getting rid of CCR5 makes certain cells more susceptible to infections by things like West Nile virus. So that's a problem. What's the balance, and who gets to pick? And so we know that just like a butterfly flapping its wings in Brazil, causing a hurricane in Borneo, that very small changes can have very large unintended consequences. And so I think a very natural thing to say as a human is, let's not do anything until we understand everything. But I would argue that making that argument now is cutting us off from a future that we can design and get to, in which we can reap the benefits of these types of technologies while still holding on to the things that we think are really important. Rather than refuse to use it, we need to decide now what's important, and we need to design our tests and our strategies to make sure that we're holding on to those things that are important. So who gets to decide? And the answer is us, every one of us in this room, because there's no putting this genie back in the bottle. This isn't something that we can say, oh, my kid will figure it out. It'll be fine in 30 years. Time's up. Now's the time. And so what I'd love for you guys to do for me, in fact, is share your voice with me about what it is that you think about the things we just talked about today. So go ahead and grab your camera and take a picture of this slide, so at least you have the URL. And that was the best looking URL I could get you. I'm so sorry. But I also made a QR code for you. There's 7 very, very short questions in this poll that are going to help me understand what you think about everything we just talked about in terms of your values, your morals, and your ethics. And this is all important because at the end of the day, we are talking about human beings. We're talking about the future of humans, not just individuals, but a species as well. And I loved this quote that came from the Bioethics Committee that says, "We are human because of the interplay of many biological, historical, and cultural determinants which preserve the feeling of our fundamental unity and nourish the richness of our diversity." Edited humans are still humans. You would have to have tons of changes in order to actually get a different species. That's not what we're talking about here. We're talking about designing a future that both honors the diversity of human DNA and humans as a species, but finds the way to bring us to our best potential and hopefully abundant health. That's where I'm going to leave us today. Thank you so very much. I'll talk to you soon.